Okay, thanks, Raja, for being here. The floor is yours.
Perfect. Thank you very much. So it's really a pleasure to be here to to talk to all you guys and to share the work that we are actually doing. So what I'm going to be presenting, I'm the only person presenting here, but please do keep in mind I have a ton of people to thank here, all the members of the OligoArchive consortium and all the collaborators outside, some of whom I will mention later in the talk. So today I'm going to talk about a problem that has quite often been overlooked by by many communities, right? So, and we're going to talk about storage problems. So the setting that I actually want you guys to have in mind is how do I actually store data for the next 100, 200 years, for example, right? If you just start down that thought process and start thinking about everything that could go wrong, it will very quickly lead you into several issues that we have with today's storage hardware. And what I'll go in today's talk is I'll show you how these issues are not hypothetical. So something that only you might dream about or you might think about, but these are actually real issues that are affecting real companies, real industries today. And to solve these problems, we need something quite radical, right? So it's no longer enough for us. Current technologies are no longer going to cut. We need something quite radical. So the title itself will give you an idea. So we are going to be looking at storing digital data. But normally digital data, we store it on digital media, right? But instead, what we are going to see is, can we store digital data on analog media? I'm going to show you some things that you would not think of as storage. For example, something like this, which is a piece of paper, right? So would you think a piece of paper as storing data? No, I mean, normally you wouldn't, right? But I'm going to show you why you might want to think of that. And similarly, storing data on biological media, which is DNA. So I'm going to walk you through these things in this talk. So at first,
Raja, I just want to say that up until 50 years ago, we did think a piece of paper as a way to store media, right? To store data.
Indeed. So we would consider analog media for analog storage, right? So you would think of paper for storing news articles or handwritten things, or even today government gazettes, all the records are in written form, right? But if you think of digital data, would you think of storing the JPEG photos that you have on your laptop, for example, right? Would you think of storing that on paper? Not in a printed photo form, but maybe in a more compressed form. And I'll show you there are different ways you can actually do it and why you might want to do it, right? So that's a good point.
That's great.
Yeah. So it's a good point. Yeah. So the context here is that, as all of you know, I am preaching to the choir here. So everybody is generating data, especially given the boom in machine learning and AI and data analytics, right? So IDC is predicting that by 2025, we are going to have 175 zettabytes of data sphere. That's the sum total of all data generated worldwide. That's a huge amount of data, right? And 50% of that data is expected to be enterprise data. So data that sits in data lakes, data warehouses, databases, name whatever you want, right? Now, what's interesting about this is that not all data generated is going to be hot. You guys are actually pretty much working on the bleeding edge of hot data, right? So you are at the hottest end of hot data. I'm going to move very, very, very far away from you. Okay? I'm going to basically move to the other end of the spectrum, where I'm going to look at data that has been generated and data that's not going to be used very often. Okay, you are all looking at data that's being used in microseconds, milliseconds. I'm going to go further away. Why am I looking at this? It's because this data, which is referred to as cold data, just data that's generated and infrequently accessed, constitutes about 80% of this total data, right? Of all the things that we are generating, 80% is going to be cold. And this is growing at an alarming rate, 60% per annum, 60% cumulative annual growth rate, CAGR. That's what I'm showing here, right? So this data is growing rapidly. It's a vast amount of data. And if you look at where are we going to store this data today, how are we going to store this data today? I'm showing you here all the storage technologies we have available today. You guys already know all of this. We have DRAM with nanosecond latencies, SSD with microseconds, hard disk drive with milliseconds, and tape with minutes access latency, a few minutes of access latency, right? And I'm showing you here that, as you guys already know, the storage cost is going to increase as you go from tape to DRAM. So we are going to walk from the archival tier today, which is made of tape. Then you have the capacity tier today, which is made with hard disk drives. And you have the performance tier, which is made of DRAM and SSDs. You guys work in this performance tier. I'm going to go all the way down to this archival tier, okay? And the problem here is that if you have 175 zettabytes of data, 80% of it being cold, okay? A vast majority of it is cold. All of this today has to be stored on tape. Obviously, because one, the data is not being accessed frequently. So it makes no sense for you to store it in a random access device like hard disk drive or SSD, right? It's not accessed frequently. So you store it on tape, which is the lowest cost, cheapest storage medium that we have available today. But the problem is that current tape-based archival system suffers from fundamental limitations. Okay, for those of you who are not aware, because you don't work on this, tape is a multi-billion-dollar industry on its own. Okay, many people are surprised to hear when I tell them that tape is used today. It's very much alive. It's used widely. I mean, almost all cloud providers use tape. Almost all large data storage installations use tape. So tape is widely used. It's as I said, it's a multi-billion-dollar market. And current tape-based archival has fundamental limitations. Okay, so what are the limitations? So the first thing about long-term data storage, the data that's being generated is a survey from SNIA, the Storage and Networking Industry Association. They did a study called the 100-Year Archive. So what they did is they went to many different companies, archivists, museums, enterprises, whole bunch of people, and they asked them the question: if you're all gathering a lot of data, right? How long are you storing this data? And it turns out 60% of archival data or cold data, right, is going to be stored longer than 20 years. So most people said, we are gathering this data, we are using this data, and we need to store this data because we have to meet certain legal compliance requirements. So somebody later could come back and sue us, and we need to be able to produce the data. Or we need to meet certain other, if they're health-related institutions, they need to preserve a patient's data until a few years after the patient is dead. For example, that's like already a 80+ year time span or 90+ year time span, right? So many of these cases, or if you're a museum or an archive and you're preserving some digital art, you have to essentially preserve it forever, right? The data does not have an expiration date. You just have to keep it forever. So increasingly, a large number of people in various contexts, they are facing the situation that they have to preserve data longer than 20 years. And this is the starting point of the problem. If you look at the current technologies we have available today, I'm showing you the SSDs, hard disk drives, and tape. And if you look at the durability, on average, how long do they actually last, right? It turns out media decay is a major issue, the first major issue. Tape, which is,
A quick question. So this media decay that you mentioned, is it like if I take a flash and I put it in the shelf and it works, right? The moment I do that, and there's no...
Yes, you just let it sit.
Yeah, this is, it might not work after 5 years. Even
exactly.
Though, okay.
Yes, so it's, I'm putting ballpark numbers, right? So it's not precise. Yes, it's of course possible to build flash, which has a much longer endurance, let's say 10 years or 15 years, right? And hard disk also, it's possible to build hard disks which have 10 years. Tape vendors can show now they can build up to 20 years maybe, right? But that's pushing the limit. Once you go beyond it, all of these are magnetic media and they all rot. So like, they all have issues and they all lose data one way or the other.
Right? So we need to do the equivalent of refreshing memory cells every millisecond or nanosecond. We need to do it for our archival storage, right?
Exactly. So that's exactly where I'm getting at. Yes. So the first problem for the refreshing is media decay, right? So you have you keep something, it's just going to go away. The second problem associated with storing data for longer than 20 years is what's called media obsolescence. You might say, okay, you know what, 10, 20 years is long enough, right? Okay, I'll actually store it. It's not a problem. But wait. You can't store that. There is a reason. The reason for that is because of the table that I'm showing you here. Now, for those of you who don't know, tape, actually, there is an organization called Linear Tape Open. Okay, so LTO, they actually put out a standard for tape storage every few years. And LTO is the standard by which tape from all major vendors basically gets upgraded. Okay, so what I'm showing you here is generations of tape, LTO2, LTO3, LTO4, so on. Associated with every generation of tape, there is a tape reader. So there is the tape cartridge itself, which contains the actual tape. And then there is a tape reader, which can actually read data off of the cartridge. For those of you who are from my generation, if you have actually used a tape cassette, you know what I'm talking about. Okay. If not, just think of the same thing. It's the same thing with the hard disk also, right? So you have a platter, that spinning platter that has the magnetic media, and you have a head that's going to read data off the platter. It's the same idea. Now, the thing is, you have to use a head that's matched together with the medium. Okay. So if you're using a LTO6 tape drive, you can actually read data from LTO6 tape, LTO5 tape, LTO4 tape. 3 generations you can go back. When you use the latest drive, you can go back 3 generations, but you cannot go more than that. Okay, if you have a LTO3 tape, you cannot read it with the LTO6 drive. And LTO is marching ahead every 3 years. Okay, so every 3 years, you're going to have a new generation of tape LTO, which means you're going to have a new LTO drive. LTO drive, new generation, typically bumps up the bandwidth by a factor of 2 or by a factor of 3 or whatever. And you also get a bump in the capacity by a factor of 2. So there is natural incentive for you to move from one generation to the next, right? Because when you, within 3 years, you get a doubling of capacity or a doubling of bandwidth, which is very, very good. But the problem with this is, as you go from one generation to the next, the drive is going to retain backwards compatibility only for up to a, two generations back, right? This is actually the second problem. The problem is today, all major tape vendors work this way. If you have a later generation LTO9 drive, you can only read up to LTO7. You cannot read beyond that. So now if you have data that you're storing in tape today, you have to be able to see, foresee it. Within 20 years, more than 20 years, you will actually have a new generation of drive, which will simply not even be able to read your tape, right? So this is the second problem that we have today that's going on in the industry. This is the media obsolescence issue. Even if you have the media perfectly valid, sitting there, you can read it, you won't have a reader to read it, okay? You will have to hack something up yourself, trying to fix how, take a drive which has been patented by one of the companies, hack it up and come up with a solution that works for an older tape. Archives are actually doing this today, national archives, right? They are trying, because they have tape from long back, they're actually doing this today, but it's a painful process and nobody is going to help you because there is no monetary incentive, incentive for this, right? So this is what's happening, okay? There's the second problem. Then there's the third problem. Forget these two problems I told you that I mentioned, okay? You have stored the data today in your favorite database, whether it's Postgres, MySQL, it doesn't matter. You have stored the data. Now you want to, you have generated a lot of cold data. So what you're doing is you can use Postgres to dump data out into an archive. For those of you who have done this, you know what I'm talking about. So you essentially extract out the cold data from the database and put it in an archive. The archive file has a particular format. Okay, you can actually choose to put the archive file in a text format, but it's going to be super bloated. You're taking binary compressed data and you're converting it into text. That's you wouldn't do that. Okay, so you can store it in a binary format. But binary format means it can only be understood by Postgres 7.8.9.10. Okay. If 20 years later you come up with a different version of Postgres, what's the guarantee that this particular binary format will even be recognized by that Postgres, right? And it turns out that we did a very quick study. Many binary formats are not recognized by their own databases simply 2-3 generations later, because database technology advances so quickly, right? Thanks to people like you who come up with new indexes and new layouts. Database technology advances so fast that databases are themselves they don't want to keep compatibility with their layouts that are 10 years or 20 years older, because that would mean a lot of redundant code, right? So this is the third problem, is format obsolescence. And it's a bit broader. It's not just specific to databases. You can also ask the question, suppose I store any file in a very specific format, how do I make sure that I'm able to access that file 100 years later? How do we know that JPEG is even going to be there 100 years later, right? So all these questions you come up, and this is related to format obsolescence. So now you actually have these three horsemen, right? You have these three things that are going terribly wrong in terms of long-term storage. You have media decay, current media obsolescence, particularly with tape, which is the only media option, frankly, that's available today. And you have format obsolescence, where you have no guarantee that data can be recovered. Put these three things together, and I'm showing you here an article that was published in 2017 by Marty Perlmutter. You can go and read this article if you look at the title, okay? The point is, Hollywood movie archives they have to preserve movies that are made in the Hollywood, right? And increasingly movies are shot with 4K quality. Increasingly movies are actually much longer than what they need to. So the 3-hour movie is what you see, but when it's actually shot, it's much longer. The editors actually clip out a lot of stuff, right? And the directors now, they want to have everything, because they want to have the freedom during editing to decide what goes in and what doesn't go in. So a typical movie can easily run into hundreds of gigabytes or terabytes even, right? One movie. Now, Hollywood archives have to preserve this content for posterity, right? How are they going to do this? So far, Hollywood archives had been relying on reels, actual film reels. But since tape technology came about, and since we started making movies in a digital way, archives started using tape. And when they started using tape, they actually realized that with media decay and media obsolescence, they have to migrate from one generation of tape to another every 5 to 7 years. Okay, the tape vendor comes to them and tells them, here is a great new generation of tape, migrate, otherwise your tapes will no longer be readable within 5 years. So they have to migrate, and this migration itself is very, very expensive. It costs millions of dollars for them to migrate from one generation to another. So what's the solution today? The solution is they're simply not preserving everything. Okay, so archivists have openly said, Hollywood archivists, that there's going to be a very large dead period from 90s to 2020, which is the period that they were looking at, where most media, independent media that's produced, not the mainstream media, but any independent films that are being produced by, people who are not super famous, right? They will all be lost. They will not be stored. This is not a problem. We think it's not a problem that's hitting Hollywood. It's hitting Hollywood now because they have the pressing need to keep it. But we think wait for a few years. And I think that the rate at which we are gathering data, I think pretty much all enterprises are going to face this kind of a problem. Anybody who's using a very large data set, right, for machine learning or whatever it is, right, they're storing data.
So Raja, I have a related question. I mean, first of all, I totally see this problem, but when I think about this problem, I also think about the problem of, do we have to maintain everything, right? So this is a different thing, that not exactly the one that you're addressing today, but maybe we can discuss later at the end if you want, or if you have a thought now, I would be happy to hear it. What would be some ways of actually automatically or not automatically, but gradually summarizing some of the data? I know maybe this is not the perfect example, the movies, even though it is also a good example. Maybe after 50 years of a movie being shot, you don't need to keep everything. You can keep, I don't know, 6 hours instead of 3, right?
But no, that's there. I think it's definitely a line that's worth thinking, right? And I think basically when this is the job of if I remember right, there's this distinction between a librarian and an archivist. Okay, and one person is basically wants to keep everything, and the other person wants to remove as much as possible, right? Get rid of the stuff that they don't need. Okay, and this is certainly a line of thought, right? So first of all, I agree with you.
I'm getting into something that essentially it's not about the Hollywood data. I'm getting into something else. Can we also, maybe in conjunction with what you're doing, right? Can we also have automatic processes that essentially archive the data in that way, that summarize the data? And if you have lots of simulation data from the Large Hadron Collider, right? Do you keep everything? Do you summarize them automatically in a way that you keep the gist for the?
Yes, absolutely. I think that's definitely there. So there are two associated problems. One problem is the quantity of the data, right? And absolutely. In fact, we ourselves, actually, I'll tell you about this. I'm not going to talk too much in this presentation, but there are compression techniques that you can actually use to summarize the data, right? And in fact, one of the things we are doing is precisely that. So if you have a large collection of images, which you are, for example, using for machine learning training, and you want to basically train a neural network for making classification based on that, right? So you don't really care about the images themselves, as long as the machine learning model, when it's trained with these images, it produces the correct classification. If that's the case, can you, instead of storing the images, can you store something else, right? That's representing the images, but maybe consumes 100 times or 1,000 times less space. And so we have this line of work here where we are looking at variational autoencoders, a specific type of neural networks that allows you to sort of extract latent space representation from images. And instead of storing the images itself, we're actually going to be storing the latent space, which can be 100 times or 1,000 times smaller, right? And this is exactly the rationale, what you mentioned. So it's about how do you use the data, right? So in terms of the quantity. So yes, there are definitely interesting tricks there you can do. And I think there's a lot of open questions there, right? So the second aspect associated with it is the long-term storage. So you might have, and I will give an example of this later in this talk, where you have certain data that doesn't matter, it's not too big, it's big enough, it's not too large, but you have the requirement that you have to keep it definitely for 100 years. There's no escaping that. Then what do, right? You're still stuck with the same limbo. You're still stuck with the same problem, right? And so I think this, basically these two aspects are there to what you mentioned. Yeah, I hope it's okay.
Yes, perfectly. Yes, makes perfect sense. Yeah.
Okay, so then let me introduce what we are looking at, right? So we're looking at DNA as a digital storage medium. So the DNA that I'm talking about is exactly the same DNA that all of us have in our bodies. That's exactly the same DNA that nature has actually used as the hereditary, the medium of transmitting hereditary information, right? It's the same DNA. So it's a small biology primer for all of you. DNA is a macromolecule. It's a molecule made up of molecules. These submolecules are what are called nucleotides. And there are 4 types of nucleotides. You have adenine, cytosine, guanine, and thymine. Okay, ACGT. A DNA is basically a linear sequence of these nucleotides. So you take each nucleotide, you attach it to another, and then you form a chain. And that's basically what a DNA strand actually is. In the human body, the DNA is a twisted double helix. So this is very, very famous. The right-handed helix that Watson and Crick invented. So we all have heard about it. When I talk about DNA, I'm not talking about this biological DNA. Okay, I want to make this distinction. I'm talking about synthetic DNA. Over the last 30-40 years, chemists have perfected the way a mechanism to make this DNA, to build DNA outside the human body. Okay, this is exactly the same DNA, exactly the same nucleotides. You can actually use a chemical process to stitch DNA outside the human body. And when I'm talking about DNA, I'm talking about this DNA that's actually synthesized outside the human body. It has nothing living to do with it. Okay, we're not going to put this DNA in a cell. We're not going to do anything, that's a living of that sort. And the second big difference is the DNA that I am talking about is a single-stranded DNA. It's not double-stranded DNA like the human DNA or like the biological DNA. Okay, it's a single strand, one strand. So now, before I tell you why we look at DNA, the general procedure that we want to adopt, I just want to share it with you so that you get a rough idea. So the general idea is you actually take some binary file, okay? You take the binary file and then you encode the binary file and you converts your sequence of bits, zeros and ones, into this sequence of nucleotides, which is a DNA, okay? I'll go into much more detail soon, but it's just giving you an overview here. Once you have generated these sequence of nucleotides, you can then synthesize this DNA and manufacture actual DNA outside in a test tube. Okay, so you can basically put DNA in a very, very tiny test tube. And once you have it, you can sequence this DNA back to get a bunch of what's called reads. Okay, and this technology is exactly the same that's used for sequencing for example, the human genome, the human DNA. So whenever somebody has cancer and they actually, now we are talking about precision oncology for treating cancer based on a person's genome. For that, they would extract DNA from the patient and they would sequence the DNA to identify what the genetic makeup of that person is and what mutations are actually causing the cancer. And based on that, they provide treatment corresponding to those mutations, right? So that's what precision oncology is. And the sequencing technology that's used there is exactly the same that's used for reading synthetic DNA, in our case, for data storage. And then from the set of reads, which is basically an approximate representation of the original characters here, I'll show you more later. We then decode the data to convert from these characters back into bits. Okay, so that's the rough outline of how DNA storage works. Now, why do we want to look at DNA? There are several reasons. I'm going to list the most important ones here. The first reason is that it's an incredibly dense 3-dimensional storage medium. Okay, so I'm showing you here a graph from the Semiconductor and Synthetic Biology Consortium. So they actually have a roadmap, they have a documentation, and I'm showing you from there. Oops, sorry. I just jumped ahead, I think. Yeah, so what this graph shows you is it shows you the density that's of various storage media that's available today, the projected density into the future, and the density estimated for DNA. Okay, so you can clearly see DNA is already 7 orders of magnitude denser than tape. Okay, so it's a huge difference that we have already with DNA, huge advantage we have with DNA because it's a very, very small. Yeah.
So, sorry, I have one more question. So here I observe, before we talked about DNA, I observe that flash has, according to that, much higher density than hard drives.
Yes. That's, I think it's, so once again, this is the volumetric information density, right? So if you have been following flash evolution, it's because of the fact that with 3D stacking, they're actually able to increase the density much faster than hard disk drive.
But so yes, I just wanted to follow up on that. So how about, but this hasn't yet reflected to the cost per gigabyte, right?
Exactly. Yeah, I think I know where you're going. You want to ask, can flash replace hard disk as a?
I mean, it's not the question of the talk, I know, but essentially, from if the flash trends continue, maybe there is also a resource limitation / cost. You know, silicon is going to be it might be a scarce resource in the near future. Who knows, right? But anyway, so that's exactly what I was thinking.
No, absolutely. I think this thought has already crossed the minds of several storage people, right? So I think even like maybe 5 years ago, 10 years ago, there was an article from, I think, Ethan Miller from UCSC, I think. So they had an article where they looked at using Flash for actually archival storage, not even capacity storage, right? So it's really, can Flash be used, just as a layer above tape, right? And does it make sense? And they were making a case why in the near future it might actually become feasible to do that, right? So it's definitely there. I mean, it's a competition between these things. It's hard to decide how things go, because now you probably saw, like, with hard disk drives, they are working on single magnetic recording, right? So there's like, they're trying to push the limits there. And then after that, there's supposed to be heat assisted magnetic recording, a few other technologies, but I don't know to what extent they have a roadmap ready for tape.
I mean, for storage, I think they are, sorry, for disks, for hard drive storage, I think they are pushing the envelope exactly because Flash is pushing the envelope and they need to compete, like the disk vendors and the Flash vendors.
Yeah.
But as you said, and then this is again not the topic of the discussion, but as you said, all these other technologies for disks actually they are making disks something different than what they used to be, somewhat different. So they have a lot of not problems, but they are opening also new challenges, right? So it's not a one-to-one mapping.
Indeed,
in the lower level, of course. In the high level it is, but in the low level it is not.
Yeah.
Anyway,
so I don't think I have that here, but I think since you are working on storage, right? I mean, you guys must be aware of the Crides rate. So there's a very important rate which is called the Crides rate, which is how quickly does the storage density actually improve. And if you look at that, hard disk drives traditionally they had 40% improvement per annum, which is faster than Moore's law, right? But what's happened over the last 5 or 10 years is that the Crides rate has dropped below, I think, 20%. Now it's like 15% or maybe even 10%, something like that. Flash and tape, they are continuing at 33% per annum and 40% per annum. So these two technologies, at least over the last 5 years, they have had no problem in scaling the density especially because of these volumetric techniques where they're able to stack things one on top of the other. And tape has no problem. They still have a lot of room to go, right? So these things, it's interesting.
I think the problem with the disks is that they are set between archival and fast storage, and essentially they are very much limited by the physical movement, and the physical movement is a huge problem. I mean, of course you can try to increase the volume, sorry, the density, but essentially, apart from that not being trivial after some point, you are still going to pay the physical movement, and
yes,
this is
absolutely
taking a big toll. I mean, for archival storage and tape, you don't care about random access so much anymore, so it's fine, right? Your sequential access is actually pretty good. So it seems to me that unavoidably so disks will have a problem, right? I don't.
It looks like that. Yeah, unless it really depends, right? Unless market forces change, it's hard to predict the pricing and what will cause what.
That's true.
Go up
That's
and
true.
down. Yeah. I think ultimately you're right. It will come down to pricing. There's no question. Yeah. So yeah, I mean, so continue just like this. The first reason is the density, right? So it's massively dense. So this is why you might have read articles where you must have heard articles which say that you can store like an entire world's data in the back of a semi-trailer in 1 kg or 2 kilograms of DNA, right? So you can imagine the density, and so that's the first point. The second point is it's durable and it's eternally relevant. So this is another interesting aspect. So there's a team of researchers from Harvard led by Professor George Church. So these guys, they are on a mission to resurrect the woolly mammoth. So what they did is they actually went to Siberia. They're around the fossils, permafrost fossils in Siberia. Around 5,000 years ago, there was the species of animals called the woolly mammoth, which is a photo shown here. The species went extinct 5,000 years ago. So these guys went there. They actually managed to extract some DNA from the fossils, from the remains, and they used the DNA together with gene editing technology like CRISPR. They used it to edit the genome of an elephant in order to try and see if they can actually bring the woolly mammoth back to life. And actually, a couple of months back, or maybe a few, I don't remember exactly when now, but very recently, there is a company that has been established on this. If you want, you can go and look it up. And their mission is to actually what they call de-extinction. It's taking species that have been extinct and they're trying to bring them back to life using this very technology that I'm telling you about. Okay, and the key takeaway from this is basically the point that DNA is durable It's very durable. So in this kind of environment, if it can last, if you can keep DNA in a normal temperature, room temperature, normal conditions, it can last forever, like hundreds of thousands of years. It can definitely last, and it's eternally relevant, because as long as we live on this planet, as long as we live anywhere, as long as there's any life, right, we will have the need to sequence DNA. It's just a given. I mean, biology now, has been radically transformed because of the whole thing with the human genome experiment and all that, right? So we have the requirement to sequence DNA, which means anything that's stored in DNA will always be readable in the future. We will always have readers. Okay, there's no problem with that. There's also a very interesting aspect here. Unlike all the storage technologies where the reader and the media are coupled together, so the tape reader and the tape media are coupled together, right? Same thing with the hard disk. DNA separates out the two. So DNA as a medium itself, if you store data on DNA, you can use any sequencing platform to read the data back. And we have already gone through 3, 4, 5 generations of sequencers in the last 30, 40 years. And DNA works with all the generations of sequencers. It's independent, right? This gives you an added layer of eternal relevance, so to speak, right? So we don't have that particular problem that tape or current medias have, which are tied to the reader. So based on these advantages, we actually started a project, a European project. It's a future and emerging technologies project called OligoArchive. And the focus of the project was to use DNA as an intelligent storage medium. So we wanted to build a full storage stack based on DNA. So starting from the bottom, where we have the media layer, where we want to look at synthesis and sequencing. So think of this as a hard disk. So inside the hard disk, you have some synthesis and sequencing machinery that can write DNA and read DNA, okay? And on top of that, you have a controller layer where we can do near molecule query processing. I'm not even going to talk about this because I don't have time today, but Thomas Heines from Imperial, actually his group works on this. The idea is, once you have stored data on DNA, can you transform certain operations that you do in silico, meaning you do it in a computer, in your PC, right? Using your processors. Can you transform those operations into biochemical reactions that are done directly on the DNA? For example, we had a paper, this was one of the first papers we published in CIDR, where we actually showed that you can actually do a select query or a join query, maybe even, right, to a certain extent, directly on DNA. So you don't extract the data out and then do the processing in your processor. Instead, what you're going to do is you're going to directly transform these operations into biochemical reactions that run on DNA and produce the result, right? And the reason why you want to do this is because DNA, as I told you, is not only massively dense, it's also massively parallel. So the biochemical reactions can happen on multiple strands of DNA simultaneously. And this is why you want to do it, because it's massively scalable. And there is a lot of, this line of work is an entirely new line of work that people are investigating, where they're looking at using DNA as a computational medium, right? So I'm not even going to touch this in this talk because I don't have the time. And then we have the operating system layer, where now you have a DNA disk, which has a controller and media, and the OS layer is basically responsible for providing file system abstractions or block abstractions or whatever it is, right? And this also turns out to be non-trivial. And finally, you have the application layer, where you figure out how do you encode your database, or how do you encode images, videos, whatever it is. So these are the layers. So this is what we wanted to look at. And in a consortium, we have a group of people who have expertise in each layer, and we are basically putting together all of this. Okay. So in our terms, for our case, for this talk, the architecture that I have in mind for DNA storage looks like this. So you have a DBMS. Okay. There are going to be two utilities, archive and restore. And instead of dumping out a binary file, what these two programs are going to do is they're going to take your database that's stored in the DBMS, and they're going to dump out a sequence of characters like this, AC, GT, GAT, whatever, right? These are the actual DNA strands that need to be synthesized. Okay, so you're going to take the binary data and print out a bunch of strings like this. And these strings are going to be sent to a DNA storage system, your DNA disk, which is then going to use a synthesizer to synthesize the DNA and store the DNA. And later, when you want to read the DNA back, you pass the object identifier. So you have an OID associated with every table or every column or whatever it is you want to store, right? So then you use the object identifier, you pass it back to the disk. The disk will basically use a PCR thermocycler. So this is something that amplifies the DNA. It's the same thing that's basically used when you do the RT-PCR test. So the viral RNA is amplified. It's the same technique. So the PCR thermocycler will amplify the correct DNA that you want, and the sequencer will then sequence the DNA and bring out a bunch of strings. These strings are going to be different from these strings. Okay, I'll get back to that in a minute. And then you pass these strings to a restore utility, which will restore the DBMS back. So it's the architecture. And you see this nicely fits into the existing DBMS, right? So you don't have to do, all you have to do is write these two programs, and then the rest of the database doesn't even have to know about anything. Okay, so that's the overall architecture. Now I want to talk about what are the challenges in storing data on DNA. Okay, so there are a bunch of challenges. All DNA is all great. I mean, you know, the benefits, but there are many issues. The first issue is each DNA is limited to a few hundred nucleotides. So, and I mean, if you take the DNA as a strand, you have A, G, T, C, and so on. You can only have up to a few hundred of those nucleotides due to the limitation of synthesis that we have today. Beyond that, you start getting many errors during synthesis. So when you manufacture the DNA, you will start making many more errors. Okay. So the important thing is when you store data, so if you are looking at, let's say 200 or 300 nucleotides, each nucleotide can store basically 2 bits of information. Why? Because if you think of it, at every position, you have 4 possibilities, A, C, G, or T. Okay. And so with 4 possibilities, you can store 2 bits of information. So a 300 long nuclear DNA strand can store about 600 bits of information. That's it. So on one DNA, you can store a maximum of 100 bytes. Okay. That means if you want to store any reasonably large file, you need to store it across millions of strands of DNA. Okay. That's the first thing. The second thing associated with it is that not all DNA is the same. So you cannot create a DNA that looks like A in all A's. Okay, that's an invalid DNA. You cannot synthesize it. Current synthesizers are not able to do that. Current sequencing technology cannot read it. Okay. In fact, DNA has certain biological limitations that you have to obey. The number of repetitions of a nucleotide should be capped. It cannot be more than, let's say, 4 or 5. You cannot have 5 consecutive A's or 6 consecutive A's. The synthesis machinery will not work. Okay. What we call the GC content, which is the ratio of G's and C's to A's and T's in the DNA. So you manufacture DNA, you cannot have 80. No, that's not a valid DNA. Okay, because the GC ratio is basically 0. There is no G's and C's in the DNA. Similarly, you cannot have a DNA that GC is 100% GC. So all these biological constraints are there. I'm naming a few here, but you have to obey these constraints when you generate the DNA. Okay. And finally, DNA has no addressing. So once you generate DNA, it's going to sit, it's a 3-dimensional storage, as I said. It's basically sitting in a pool where it has no addressing. Like unlike a hard disk where you can say, I want block number 0, block number 1, block number 2. There is absolutely no way you can fetch one piece of DNA by saying, I want that DNA number 1200. Doesn't exist. Okay. Which means you need to add addressing on top of the DNA. So you take the file, you break it up, you have to add addressing yourself. Okay. So that's what you have to do. So when you encode the data, you have to make sure that all of these conditions are actually met in converting the binary file into the sequence of DNA strands. Okay. Then you synthesize it. And after synthesis, you're going to end up storing the DNA in a test tube like this. I'm showing you the test tube that I received back from the synthesis company for the very first synthesis we did. This is a Postgres database that was stored in DNA. And it's like, you can see it here, it's like 100 nanograms or something like that. So we got it. And now when you want to read the DNA back, you have to sequence it. Now the sequencing process does not give you the original strings you generated. Okay. The sequencing process introduces biochemical errors on its own. The synthesis process will also introduce biochemical errors. Every string that you gave in will be replicated multiple times, and each replica will look different. Okay. So these are noisy copies of the original. So I'm showing you here is a green original string. You have green copies, 3 green copies. So you see some copies have errors, some are okay. Then you have the blue string, which is replicated twice, and you could have other strings which do not appear at all, which are completely missing. Okay, that's basically due to bias in PCR reactions where you amplify the DNA. Some DNA is amplified a lot and some is not amplified at all. And sequencing can miss this out. So now you have your original strings completely gone because you have stored it in DNA. That's the whole point. You cannot keep these strings, because if you can keep these strings, there's no point in storing it on DNA, right? So you stored it on DNA. Now you try to read it back. You get this jumble of strings. No ordering. It's up to you to figure out what the original strings are. Okay, so you need to do some kind of a consensus approach here, which figures out what the original strings are given this noisy copy, and then you apply decoding to recover back the original data. So that's the problem.
So Raja, sorry, to understand something, maybe you said and I missed it. When we're doing the synthesis, I mean, there's probably a lot of questions that are going to be answered later, but one question: are we going to generate multiple copies of the same strands just
Yes.
so we have redundancy? Yeah.
Yes, absolutely. So in fact, the synthesis process itself will, depending on the synthesis process, so some synthesis process can generate multiple copies of the same strand. Some can actually, and these multiple copies need not be the same length also. Okay, so some of them will be, you will get this physical redundancy where some copies are all identical, some copies are very short, some are very long, because the synthesis cannot, be very precise there, right? So you can definitely add some copy noise there, and then you will take this DNA, you will use PCR to amplify it, you will make multiple copies of the DNA before you sequence it, then you sequence it, it will add more errors to it.
Makes sense.
Yeah.
Okay.
So that's
it's very fascinating to see all this pipeline. I probably have more questions, but I'm gonna wait until you say more before I ask any.
Sure. Yeah. So that's the point. So to basically solve all these problems, and to what we have to do in OligoArchive is basically come up with a pipeline that can solve all these problems, right? You can store the data and still be able to decode it back. So that's what we have done. So I'm going to give you a very high level overview of the pipeline, and I'll tell you why. I cannot go into details because some of these components are like full elaborate works on themselves. Okay. So for example, what we do is we take the binary file. Then the first thing we do is we actually randomize the binary file. So you use the pseudo random number generator to mix up the bits. We do this because there is a biological requirement that if you have multiple strands of, so let me go back. If you have a sequence of bits which are all similar, let's say a sequence of zeros, you have 1000 zeros, okay? When you break it up, you will get 100 zeros, 100 zeros, 100 zeros. And all these will end up generating the same DNA strand, right? When you have multiple DNA strands which look identical, there are problems that can happen in sequencing. So that's one thing. The second problem is that when you have multiple strands that are identical, during the decoding part, it becomes very difficult for you to infer how many of these strands did you generate in the beginning. Remember, you're not storing any information, right? So in order to basically avoid all these issues during decoding, what we do is we randomize the data. So to maximize the chance that two DNA strands don't look similar to each other. That's the first thing that we do. And this helps a lot during decoding. And once we do that, second thing we do is error control coding. Okay. And this is a whole line on its own. So what we are using now is we are using something called low density parity check codes. These are codes that are actually used in the 5G standard for telecommunications. So when you send message, when you use data on 5G or when you talk on 5G, LDPC is the code that's actually used for guaranteeing error control. The reason why we use error control is I told you that there can be errors, a lot of errors in the synthesis and sequencing, right? So when we store the data, we send in some strings. When we get it back, we do the consensus and try to infer the strings. But there is absolutely no guarantee that we will be able to infer everything exactly. We might have some errors during the inferring procedure. So in order to be able to recover from those errors, we add additional redundancy. Okay, for those of you who are familiar with Parity, Reed-Solomon codes, things like this is exactly what we do. So we add redundancy to it. And for redundancy, as I told you, this is a whole line of work. You can use in coding theory, there's a whole lot of literature on how can you actually, what kind of codes do you use when, right? It turns out, so to simplify things, in case of DNA storage, we can actually have, as I told you here, you can have substitutions, insertions, or deletions. So you can have characters being swapped, you can have new characters being added, or old characters being deleted. This particular channel, the DNA, viewing the DNA as a channel, communication channel, it's an absolutely nasty channel. Okay, most coding theorists, they prefer to work with only channels that have substitution errors, because insertions and deletion errors, even today in coding theory, we do not have very clear methods that give you solid bounds in terms of recoverability. Okay, so in coding theory, substitution errors are very well understood. They are very well studied. You have a lot of codes that work very well for substitution, but the DNA channel has insertions and deletion errors. A lot of recent work has actually shown that low density parity check codes is a specific type of code, which actually, when you apply it in a specific fashion using large block length, it's capable of dealing with insertion, deletion, and substitution errors. It can actually work very well with these. So what we have done is we have built on that work, we have taken that, and we are using that here as our error control coding. But we are changing it in ways that I cannot talk about it right now. It's a whole another line of work, as I said. But we have to customize it to our encoding and to our decoding. So particularly in specific, I'll just say a small thing about it. The low density parity check code takes as input what's called a log likelihood ratio, which is actually input that comes from the channel. and uses this log likelihood ratio in a message passing iterative decoder to figure out what the original bits could be. So we have to figure out how to transform the reads into these log likelihood ratios and feed it correctly to the LDPC decoder, right? So this is kind of stuff that we actually do. So we customize LDPC to match our use case. So that's the error control coding part. Then we apply indexing. As I told you, we take a file, we break it up into millions of DNA strands. We have to add numbers to each strand to be able to recover and put back the strands in the correct order, right? So we add indexing information in the strand, and we protect this indexing information using the error control coding also. And finally, we use a constraint code. Now, this constraint code guarantees that when we convert finally the bits into this sequence of ACGTs, you obey all the biological properties. Okay, so all the homopolymer, you don't have more than 5 repeats of a character. Your GC content is fine. All of these, there are no secondary structures. So all of these constraints are obeyed. So that's a constraint code. As I told you, it's a pretty complex procedure. I'm not going into details here, but if you're interested, ping me later, right? So we can talk about it more. So once we have this encoding done, we are converting the bits into these nucleotides. Then we synthesize the DNA and we store it in a capsule like this. So I'm actually showing you the physical one. This is what's called a DNA shell. It's made by a company called Imagine here. Inside this, DNA is actually stored in a dried form. It's completely dried. There is no water. There is no air. It's stored inside a glass canister inside this capsule. And like this, it can actually last for thousands of years at room temperature. You don't even need any special storage for this. And this capsule is actually storing DNA from a Postgres database right now, right? So we store it here. And when you need the data back, then you do sequencing. As I told you, you get back what these strings, noisy copies, which are called reads. You get noisy copies of the original string. From there, we have a full decoding pipeline. And the two key things in the decoding pipeline I want to mention here are one, consensus, which is a consensus procedure that we developed last year. And we model the consensus procedure actually as a string join problem. And we build on techniques from the database community actually, which look at randomized embedding algorithms to very efficiently do string joins. So to give you an example, here as the output from sequencing, you will get, let's say, 1 billion strings, okay? 1 billion strings. It's your job to figure out the original 10 million strings from this 1 billion, okay? So you have to cluster these 1 billion to generate the original 10 million. And you have to tolerate substitution, insertions, and deletion. By definition, this means you have to use edit distance as the distance metric, okay? And edit distance, if you're not aware, has a quadratic time complexity. It's extremely slow to compute edit distance between two strings. So it's not going to scale to the billion scale. So we use a whole lot of algorithmic techniques. We use locality sensitive hashing. We use randomized embedding. We used a whole bunch of techniques modeling this problem as a string join problem, where we try to find clusters of strings that are similar to each other. And we build on recent work done in the database community to come up with a consensus algorithm. That's one. The second thing we have done is we have taken this idea of consensus, the algorithms that we use for consensus, and we have actually built a tool called Axel Align that's used, that can be used in computational genomics pipeline. So when people actually read DNA from a human and they have to analyze it for finding what diseases you have, for example, the first step of that process is what's called alignment, where your DNA is aligned to a reference genome. So we realized that when we built this consensus, the same algorithmic techniques can help here. So we built a tool for that. We validated it, and we are actually using it in production in some of one of the biology labs here for sequencing real human data, right? So we're actually using that here. So these are two pieces of work that we did, and it's a high-level overview of what we did. Once you apply one consensus, you actually get as output a consensus set of strings, and then you do the reverse of what the encoding did. So you do constraint decoding, de-indexing, error control, decoding, and de-randomization to get the original bits back. Okay, so this is the full pipeline. I think it's so that's the full pipeline that we have actually developed. So
one very quick question: can you talk a little bit about the indexing?
Yeah, so the indexing is pretty straightforward. So it's not indexing in terms of the database where you have a data, you're building the index to be able to search over the data, right? So in this case, we are building an index to order the DNA strands. So all it does is you have to count how many DNA strands there are. You have to figure out how many bits you need to number the DNA strands. And you basically take your binary data and you add this addressing information before or in the middle of the binary data. That's what you do. Right, that's it. It's very simple.
And then essentially you recover that when you are calculating your 10 million strands in
Yes.
order to sort them based on
Yeah.
that, and then recreate the file.
Exactly. So we'll recover during recovery, we will get the 10 million strands. We will reorder them based on the index inside the strands. Some of them might be missing. So maybe 10% of them can go missing. It's no problem. We can recover it. And some of them might have errors, also no problem. Then once we put them back in the correct order, we pass it to the decoder, which will do the error control decoding. That's exactly the idea. Yeah.
So, and I have one question. Do we have guarantees that this will always work? I mean, there's a lot of errors and a lot of all this process. And I think it is interesting to think about the question, which the answer might be yes, right? But to think about the question, am I always going to be certain 100% that I'm going to get my initial file back?
Yes, that's a very good question, right? So in fact, the answer to that, so that's the whole thing. I think the answer to that is always yes, because if you don't care about, for example, if you don't care about optimizing the cost too much, right? You can always read back the DNA, the sequencing procedure. There is something called the depth. Basically, it tells you how many times you read the DNA. Okay, so you can read the DNA once, you can read it 10 times, you can read it 100 times, you can read it, multiple times, right? So when I read it 100 times, you get 100 strings for every DNA. So when you increase the depth, naturally, as you might expect, you're reading the DNA multiple times. And so there is a much higher probability of you being able to recover the data. And in fact, if even in our case, what we have done with some experimental stuff, right? We typically use a coverage of around 5x. It's average coverage, where the DNA is read 5 times and reported 5 times, right? And we are able to recover the data very easily. To give you an example, when you do human genome, when you sequence the human genome for various, for normal experiments, typically they use a 30x coverage. 30 times they read the same piece of DNA. For cancer studies, they use even more. They use 100x or maybe even more than that, right? So 100 times they read the same piece of DNA because they want to get very, very accurate representation of the DNA. So yes, you are always able to read the data back. So that's
I think Papon has a question.
Go ahead.
Yeah, so I have actually 2 questions. So the first thing is that can we actually read parts of the data or like partial decoding? This is one thing. And another thing is that for any type of storage media, it is actually essential to be compatible with the block IO interface that we have in our existing technology. So
yeah.
Do you have any comments about that?
Oh yes, that's both great questions, right? I think I love this. It's really a great question. So answer to both is yes, right? And answer to both is yes, we are working on it. Unfortunately, I cannot, I'm not presenting it at this slide because of the lack of time. But so to answer your first question, can we read, actually to a certain extent, both are connected. Can we read a portion of the data? That's the first question that you asked. Suppose if you had stored your data in a whole bunch of, suppose you had the block interface over DNA, you're able to address 0, 1, 2, 3, 4, right? And you store your data over a whole bunch of blocks, then that means you can selectively retrieve block number 5, right? You should have the ability to do that. So in fact, what we are doing now is in our lab is actually building a system that can provide you this block interface. So we are taking this error control logic and we are building essentially a block abstraction on top of DNA. Now, typically the way this is done, I can give you a high level idea. You take 2 very short sequences of DNA. These are called primers, priming regions. And what you do is in front of the DNA that you are generating, this is your data DNA, you attach in front and in the back 2 very short sequences of DNA that are called primers. And you associate primers together with a particular address. So when you need a particular address, you find the primers that you need. Then you take those 2 short strands of DNA, you run a biochemical reaction called polymerase chain reaction. What this does is it will actually select, given all the DNA, it's going to selectively amplify only that DNA which has these 2 short strands attached to the beginning and the front. Okay, so that's the technique that's used. It's the same technique that's used for amplifying your viral DNA, the COVID virus, right? When they do RT-PCR, this is the same thing they do. They take a lot of stuff. They take the viral DNA. By definition, by default, the viral RNA will be very small. There will be very little quantities of it, but they want to amplify it so that they can verify that it's there or not. So when they do PCR, they are amplifying the viral RNA and they want to amplify only the viral RNA. It's the same technique. So you amplify your DNA using these 2 short DNA strands, and that will pull out the DNA strands that have these 2 strands. Now, this is how you implement block addressing, right? So, and in fact, we are doing this. We are actually testing How do you design these short DNA strands to make sure that you can get good accuracy? What happens when you get, inaccurate, readouts from the DNA, right? And how can you avoid it? So we're looking at all of that right now. So it's definitely possible.
Got it. Thanks.
Yeah.
I have two quick follow-ups.
Yeah.
How much data does it fit in this capsule?
So in this capsule, I actually have to double check, but you can comfortably fit an exabyte of data in this.
Okay.
So it's a lot. Yeah.
So essentially, I was thinking that you could. I was thinking something that doesn't work, right? You could use different capsules for different parts of data, but it has to be the granularity of exabytes or something. Right?
So yes, no, actually your idea is also correct, also fine, Mana. So you can definitely do it. In fact, we are not doing it here, but in other labs, they are actually, what they're doing is they are building microfluidic chips where they can move DNA, so like in the liquid form, they can actually move it around and they can store it on plates. And they have tiny containers, you can think of it that way. And this is exactly what you're saying. Instead of this capsule, you can store it, and then you can use this as an additional form of addressing on top of what DNA already gives.
And the other follow-up is when we read, that is sequence the DNA, what happens to the medium, to the DNA itself? Can we use it again?
Yes.
Is it destroyed? Do we have to write it again?
That's a very good, yes.
Is there a read-write asymmetry, essentially?
Yeah, that's a very good question. I think also another very good question. So it's very similar to Flash, because when you read Flash, you are depleting the cell, right? So to a certain extent, you can think that you are depleting the DNA a little bit, because normally when you read the DNA, DNA is actually stored in this capsule. That's when you read it back, you rehydrate it. First, you put it in a water-based solution, right? And then you take a sample of the DNA. You don't take the whole thing. You don't take the full solution. It's not required. You only take a little portion of the solution. So there is already some left, right? And in addition, as I told you, one of the good things about DNA is you can create copies of it very easily. So this PCR reaction, you run it, within a few minutes, it's going to double the amount of DNA. Within another few minutes, it will quadruple it. So you let it run for a short time, and you will basically amplify your DNA and create many copies again. So there is no problem, right? And in fact, inside our body, this is happening all the time, right? So there are cells which are actually creating copies of our DNA continuously.
So just to verify, in this pipeline, when we are reading, it makes sense to put back a little bit of the DNA that we just recreated to read.
Absolutely, yeah, definitely makes sense. At least once in a while, it makes sense to put it back. Yeah.
Yeah, this is the refreshing, so to speak, of the medium. Okay.
Exactly. Yeah. So that's that, right? So that's the full pipeline. So what I want to tell you is, so this pipeline, I'll give you an example later, like just a quick example of what we did with it, a real use case. But we can store data with very high density on this, but it does not solve the format obsolescence issue that I mentioned in the beginning, right? So the format obsolescence, meaning you store the data on DNA, it's going to be there for 1,000 years. You can read it back, but then how do you go back and understand what data you stored, right? And there is actually a bit more of problem, a bit bigger problem. It actually complicates format obsolescence a little bit more. So when you store data on DNA, I told you need to encode the data into these oligos. Oligos are short strands of DNA, right? So retrieving the data requires converting back these DNA into digital data. So that's what I showed you here. You need to convert this back. Look what we have here. We have this huge DNA decoding pipeline here, right? And we have this algorithm here, the error control decoding, the LDPC code that I told you. It actually uses certain matrices, which are called like generator matrices or parity check matrices. It uses these matrices to transform the data. You have your incoming data, which is coming from the channel. You do some matrix operations on the incoming data, and it puts out the data that is the error corrected data. So you have to have that matrix. Where are you going to store the matrix, right? So that's basically the question. So that's what I'm mentioning here. Decoders, you need the matrix. You've stored the data on DNA. 1,000 years later, you come back and you don't have the matrix. So how are you going to store the matrix then, right? And what we want to actually do here is we actually want to archive the decoders itself in addition to the data, right? Otherwise, we are not able to decode it. And here is where we're bringing together analog media, right? So this is a collaboration together with two other people, from Vincent from Upalia and Martin from Memories of Mankind project. So if you don't know about Memories of Mankind, I highly encourage you to go and take a look. It's a fascinating project where they're looking at preserving data, culturally important data of mankind forever, right? So even in case we have a nuclear holocaust, for example, how do we make sure that we have a cultural seed vault, like the Arctic seed vault. So it's a very, very interesting project. So we have a collaboration with them. And I want to share the idea with you here. So analog media, as Manos mentioned in the beginning, it's already being used by museums and archives for storing, newspaper reels and whatever, right? So all this kind of stuff. You can store digital data on analog media. So the idea with this is to convert data into barcodes, and then you shoot the barcodes onto film. This is actually being done by a company called Pickle. Okay, this is their product. They actually have a barcode printer and a barcode scanner, and they're storing data on movie films, film reels. But of course, it's movie films that have been converted now to store digital data. They have been made a bit better, and they are competing with tape, for example, right? So this is definitely possible, and analog media kind of solves the media decay and obsolescence issue. So we know that all these media can last for more than 500 years. Paper, microfilm, film, all these can last, okay? And they require basic scanning technology. Any light projection technology, you will be able to read the barcodes off of these media, okay? But the problem is these media are very limited in density. So you can only store maybe, if you use this barcode technology, you can store maybe a few kilobytes on a single page of paper, or maybe a megabyte on a single page of paper. That's it. So if you're looking at storing large amounts of data, you still have a problem, right? So what we want to do is, I'm just showing you the bootstrap process here. I think we are basically running out of time quickly, but I'll show you what we want to do. We want to use the OligoArchive encoder to convert binary data into DNA, synthesize the DNA and store it, okay? Then we want to take the decoder, particularly the matrices, the LDPC matrices, the software that we use for decoding. We want to pass it through something that's called a universal emulator, okay? And bear with me, don't have to worry about what this is. What this is going to produce is it's going to produce a bunch of barcodes like this, okay? These are called emblems. We produce a bunch of barcodes, and it produces a bootstrap text, okay? So some 1, 2, or 4 pages of text. There is a pseudocode inside this, and then there's a bunch of barcodes. You take all of this and you print it out, or you film, shoot it out to a film, like what's shown here. It's a real film that was manufactured by one of the companies using this approach. So you can see the text on top, you can see the barcodes on the bottom. When you want to read the data back, you have your DNA stored in the capsule, you have your film. First thing, you scan the film. You separate out the text and the barcodes, okay? Then you give the text to some high school student. The text actually has 2 pages of pseudocode in English. The high school student has to implement what the 2 pages say in any language, in any operating system of their choice, okay? When they do this, they run the program, the pseudocode that they implement, the program will actually ask them for additional information. So they keep following the steps that's written in these 3 or 4 pages. And finally, the program will ask them to feed as input the barcode images in normal RGB, standard RGB form, okay, nothing special. So all the instructions are encoded there. It will take all the images, and it basically builds what we call an emulator. So an emulator is actually constructed out of this. And this emulator basically will load the decoder that we stored inside before. And the decoder will then take the reads that came in and produce the binary data out. It sounds a little bit like science fiction, but we know that we have tested it. We are actually now doing a large scale experiment. We're going to do a large scale experiment to show that it works. We know that it works with a small scale experiment. And this gives you kind of migration-free end-to-end passive preservation. You store the data on DNA, you store the metadata on analog media, right? And then we have a way by which we can actually construct these emulators. Now, very quickly, I don't want to go into too much detail because I don't think we have the time here, but the technology that we are using is emulation. It's the same technology that you used for getting the games that we had, Sega or some of the gaming consoles that no longer exist today, right? Getting those gaming consoles to work today on PCs, emulation is used. It's the same idea, but it's a slightly different concept. The concept is, in those cases, you want to get old software to run on new PCs. Here, we need to preserve current software for future PCs. So rather than emulating an old processor, what we are going to do is we are actually going to build a virtual processor with a very simple ISA that can be emulated. Okay, so it's a virtual processor that we are going to build. And the instructions that I showed you before, what the student has to write, the student actually implements the code for 2 assembly instructions, a subtract and a load. When they implement this, they bootstrap a small virtual machine. And when the virtual machine takes as input these additional things, it bootstraps a larger virtual machine, which is a 23-set ISA virtual machine. and that virtual machine then loads the decoder. It's a fairly complicated process. I'm not going to go into details here, because most of this virtual machine universal emulation stuff has been done in the digital preservation community. So there are many archives that are using technology like this for preserving their software, for example, right? So this is happening there. So what we did is we really put these two together in doing an experiment. And finally, before I finish, I just want to show you, we are bringing all of this together for a real use case, and this is the real use case. We are working together with Danish National Archives, which is a national archive in Denmark, and they have been preserving digital data since 1970. What we are preserving is actually a photograph like this, which is a photograph of King Christian IV here, he's a very, very popular king which dates back around 400-500 years. And this material has been ranked as having unique national significance. So they have to preserve this forever, right? That's what they want to store. So this image is a part of a larger collection with databases containing data about the image. There's a bunch of HTML files. There are these images, all of this. So we are going to take all of this, okay? We are taking the database that's actually stored in a standard format called CR, which is a format developed by the digital preservation community. We are going to store all of this on synthetic DNA, and we are going to come up with an analog bootstrap. And this way we are solving format obsolescence with standards. So by using CR and by using the bootstrap, we are solving media decay with synthetic DNA and media obsolescence using the bootstrap. So that's basically a quick summary of what we are doing. This is an ongoing experiment. It's not complete yet. And it's just showing you roughly what we plan to do. So we are storing 12.5 megabytes of data into 500,000 DNA strands. Okay, this will be the third largest academic storage experiment in the world. And we expect to store this in just a few nanograms with a very, very high density, right? So that's what I wanted to share. So I just wanted you to recap. Contemporary magnetic media, particularly tape, suffers from decay and obsolescence, and continuous migration is the only solution today. I told you guys today that DNA provides a biological alternative, but it does not solve all the issues by itself. And so what we are doing is we are combining standard file formats together with synthetic DNA and analog media to solve all the issues together holistically in one single solution. So that's it. And I'll just leave this open for further discussion, because I know you guys would actually have some of these questions, right? But that's it. And thank you very much. I'll take any questions that you guys have.
Thank you so much, Raja. Let's thank our speaker. Thank you so much, Raja, for this great talk. Yes, I mean, I think there's probably a lot of discussion to continue. Let me stop recording. It's fine.